Hello, I'm Dr. Zelalem Temesgen, an infectious diseases doctor at Mayo Clinic in the United States. It is my privilege and great pleasure to be able to provide this overview of COVID-19. My assumption in preparing for and providing this overview is that my audience is a broad and diverse group of medical professionals at Charles University medical faculty in Hradec, Kralove, a group of medical professionals with a wide variety of background, specialties, expertise, and even interest. For this reason, I have tried to make my presentation sufficiently informative without giving excessive details in any one area. In the future, I hope to provide further updates, including perhaps presentations focused on certain aspects of COVID-19, for example, treatment. It is hard to believe, but it was only at the end of December 2019 that Chinese officials reported a cluster of pneumonia of unknown origin in Wuhan province, China. Within a few days of that report, the causative agent of the pneumonia was identified as a novel coronavirus, designated severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. And its sequence was shortly afterwards shared with scientists across the world. The disease itself was named COVID-19. The first case outside of China was reported from Thailand by mid-January, shortly followed by cases from the rest of Asia, as well as Europe and the United States. Since then, the disease has spread rapidly and widely throughout the world. The World Health Organization declared the outbreak pandemic on March 11, 2020. As of January 3, 2021, nearly 85 million cases have been identified worldwide, resulting in nearly 2 million deaths. Not surprisingly, there has been considerable discussion and interest on the origin of SARS-CoV-2. Coronaviruses are a large family of viruses, literally hundreds of viruses, that usually circulate among animals such as bats, pigs, camels, and cats. There are 7 coronaviruses known to infect humans, usually causing mild to moderate upper respiratory tract illnesses. However, there are 3 new coronaviruses that have emerged from animal reservoirs over the past 2 decades, causing serious and widespread illnesses and death. These are SARS-CoV, the original SARS in 2002, MERS-CoV in 2012, and the cause of the current COVID-19 pandemic, SARS-CoV-2. The zoonotic origin of SARS-CoV-2 and that it recently acquired its ability to leave its natural animal reservoir, most likely bats, is widely accepted. However, no direct bat-to-human transmission has been demonstrated. Therefore, it is thought that the transfer to humans more probably took place via an intermediate host, similar to the civet for SARS-CoV and the dromedary for MERS-CoV. However, an intermediary host for SARS-CoV has not been confidently identified, though earlier reports had suggested that pangolins, which are anteater-like mammals, may have played that role of an intermediary host. SARS-CoV-2 itself is a beta coronavirus, an enveloped virus that contains a positive sense single-stranded RNA genome. It has a nucleocapsid composed of genomic RNA and a phosphorylated nucleocapsid protein, which is buried inside the phospholipid bilayers and is covered by the spike glycoprotein, which gives it its characteristic appearance. A few words about the replication cycle of SARS-CoV-2. As the first step leading to viral infection, the virus spike protein recognizes and binds to the receptor for an enzyme called angiotensin converting enzyme 2. The conformational changes in the spike protein after receptor binding facilitates the viral envelope fusion with the cell membrane. The viral RNA genome is then released into the cytoplasm and translated into viral polyproteins, which in turn are released into the cytoplasm and translated into viral polyproteins, which in turn are cleaved into smaller products by the virus-encoded proteinases. The polymerase transcribes a series of messenger RNAs, which are then translated into viral structural proteins, which are ultimately assembled into new viral particles and released from the infected cell. Direct person-to-person respiratory transmission via respiratory droplets through actions such as coughing, sneezing, or talking is the primary and the predominant mode of transmission of SARS-CoV-2. Infection may also occur if a person's hands are contaminated by droplets, for example, by touching contaminated surfaces, and then making a direct contact with the mucous membrane, such as the eyes and the nose or the mouth. Transmission from longer distances through airborne transmission, and by airborne it is through inhaling particles which are smaller than droplets, is possible, but the frequency of this kind of transmission is unclear, and it is very unlikely to be a primary mode of transmission. How does SARS-CoV-2 cause disease? The pathophysiology of COVID-19 is described as an immune hyperresponse, an exaggerated response, characterized by activation of myeloid cells and their trafficking to the lung, resulting in productions of GM-CSF and interleukin-6, further triggering a cytokine storm cascade. This in turn results in fever, low blood pressure, capillary leak syndrome, pulmonary edema, disseminated intravascular coagulation, respiratory failure, and adult respiratory distress syndrome, ARDS. Symptoms of COVID-19 appear between 2 and 14 years after exposure to the virus. The mean incubation period is approximately 5 days. 97% of individuals will have, that is, most individuals will have onset symptoms by day 11 and a half following exposure. Asymptomatic infection is very well documented in some series as high as 30 to 40%. Transmission from patients who do not have symptoms has also been well documented. The clinical symptoms of infection are highly variable. Common symptoms include fever, cough, whether dry or persistent, fatigue, anosmia, a loss of smell, dysgeusia, loss of taste, loss of appetite, and dyspnea, or shortness of breath. But many other symptoms, and some of them are listed here, are also possible, affecting virtually all organs and systems. Severe symptoms leading to hospitalization that progress rapidly to hypoxia and respiratory distress, requiring supplemental oxygen and ventilator support, are most prevalent in elderly with underlying comorbidities. Some of these comorbidities are listed here and include obesity, liver and kidney diseases, COPD, cardiovascular diseases, diabetes, hypertension, and malignancy. The most common laboratory abnormalities in patients hospitalized with COVID-19 pneumonia include the ones listed here, including leukopenia, lymphopenia, leukocytosis, elevated liver transaminases, elevated lactate dehydrogenase, and elevated C-reactive protein. Lung imaging, in particular chest X-rays, may be normal in early or mild disease. Common lung imaging findings include consolidation and ground glass opacities, and usually are often bilateral and have a peripheral distribution involving the lower lung zones. How about diagnostic testing for COVID-19? Diagnostic testing typically involves assays that detect the SARS-CoV-2 nucleic acid from respiratory samples such as nasal swabs, oropharyngeal or nasopharyngeal swabs, tracheal aspirate, or bronchoalveolar lavages. A positive test typically indicates that the patient is actively infected and is also capable of spreading the virus. Tests that detect the SARS-CoV-2 antigen, not the nucleic acid, but the antigen, may have a faster turnaround time to results. But these tests are usually less sensitive than the tests that detect RNA. Serologic tests that detect antibodies, are also available, and they usually use blood, though other biological fluids can also be used for this purpose. They have low sensitivity in the first 1 to 2 weeks after symptom onset. Their primary utility is in identifying patients who previously had SARS-CoV-2 infection, as well as patients with current infection who have had symptoms for at least 3 to 4 weeks. Let us now talk about management of COVID-19. Patients with SARS-CoV-2 are grouped into severity of illness categories. This classification helps determine the level of care needed, including the modes of therapy that is best applicable to their illness, as these categories are also utilized to describe patients participating in clinical trials of COVID-19. For example, for patients with asymptomatic or mild and some moderate cases, management is primarily in ambulatory setting or at home. This management includes educating patients on signs and symptoms of COVID-19 and its complications, and educating them that if these complications or symptoms develop, then they should seek prompt pursuit of urgent care. Management also includes treating symptoms such as fever and addressing nutrition and also hydration. And for those patients with risk factors for disease progression, then educating them how to closely monitor for those. Management of severe cases include the following: proper oxygen and ventilatory support with proper timing and use of non-invasive and invasive modalities. Routine use of pharmacologic prophylaxis against venous thromboembolism, unless there is a contraindication, and appropriate fluid and electrolyte management. While it may be difficult to distinguish COVID-19 from bacterial pneumonia, bacterial superinfection is not common or is not a common feature of COVID-19, at least early in the disease course. Thus, empiric therapy for bacterial pneumonia should not be routinely prescribed, but in practice, it often is. Once prescribed, it should be discontinued if compelling evidence for bacterial pneumonia does not emerge. Obviously, if available and indicated, COVID-19 specific therapy should be offered. This brings me to the next topic, which is COVID-19 specific therapy. There is only one drug license for treating COVID-19, at least in the United States. But there is a lot of activity in evaluating experimental and investigational agents for the treatment of COVID-19. According to one clinical trial tracker, which is illustrated here, there were over 1,200 COVID-19 related clinical trials recruiting patients at the end of December 2020. Experimental pharmacotherapies for COVID-19 can be broadly categorized as antivirals. Those are drugs with direct effect to limit viral replication, and immunomodulators. Those are drugs that limit the pathological host immune response to the virus. These experimental pharmacotherapies can be accessed through a variety of mechanisms, including clinical trials, but also emergency use authorization, at least in the United States, expanded access protocols, compassionate use programs, and off-label use of drugs that already are approved for other indications. This is a list of experimental pharmacotherapeutic agents that are undergoing evaluation. This is obviously an incomplete list. There are many more at various stages of investigation. I will give some additional information on two of the investigational agents that are now in use on more or less standard basis in managing COVID-19. The first one is Remdesivir, and Remdesivir at the present time is the only US Food and Drug Administration approved drug for treating COVID-19. It's an antiviral agent, and I would like to say a few words about this drug. First described in 2016, Remdesivir was derived from an antiviral library of small molecules which underwent intensive testing against different types of Ebola virus. Following the Ebola virus epidemic in West Africa from 2013 to 2016. It was later shown that Remdesivir has also a broad activity against a broad range of viruses, including Marburg virus, respiratory syncytial virus, hepatitis C virus, a variety of paramyxoviruses, SARS-CoV, and MERS-CoV. Demonstration of in vitro activity against coronaviruses was supplemented by data from animal models, thus supporting the consideration of Remdesivir for clinical testing against COVID-19. Remdesivir is a nucleotide prodrug of an adenosine analog. Remdesivir binds to the viral RNA-dependent RNA polymerase, inhibiting viral replication through premature termination of RNA transcription. Upon entering the target cell, it is phosphorylated into the active triphosphate metabolite, which is then integrated into replicating RNA and prevents further chain elongation, resulting in premature termination of RNA synthesis. I would like to briefly present two of the major clinical trials of Remdesivir. The first one, the one that I'm presenting on this slide, is the ACT-1, an NIH-NIAID sponsored multinational randomized placebo-controlled trial that included 1,062 patients with confirmed COVID-19 who had evidence of lung involvement. At baseline, 85% had severe disease and 27% were receiving invasive mechanical ventilation or ECMO. Remdesivir was given intravenously for up to 10 days or until death or until discharge. The primary endpoint of this study was time to recovery by day 29 according to an ordinal scale. Secondary endpoints included treatment-related improvements in an 8-point ordinal scale at day 15. As you can see on this slide, remdesivir reduced time to recovery. However, in subgroup analysis, the reduced time to recovery was only statistically significant among patients who were on low-flow oxygen at baseline. If you look at the subset of patients on mechanical ventilation or ECMO at baseline, the time to recovery due to remdesivir was comparable with that of placebo. Overall, there was a trend towards lower 29-day mortality that was not statistically significant. Among the subset of patients who were on oxygen supplementation but did not require a high-flow oxygen or ventilatory support, there was a statistically significant mortality benefit. The second important study that I want to talk about, an important study of remdesivir, is the repurposed antiviral drugs for COVID-19, the World Health Organization Solidarity Trial. This trial was conducted in 30 countries, and it evaluated remdesivir, hydroxychloroquine, lopinavir, and interferon. So these are 4 repurposed drugs in patients hospitalized with COVID-19 in comparison with standard of care. As you can see on this slide, and on the next slide, as you can see on this new slide, none of the 4 study drugs improved the 28-day mortality overall, or when stratified by other things such as age group, ventilation at entry, geographic region, or use of corticosteroids. No study drug reduced initiation of ventilation for those not already ventilated. Specific to remdesivir, comparison of the 2,750 patients randomly assigned to open-label remdesivir and compared with the 2,708 patients who were randomly assigned to standard care, there was no difference in overall 28-day mortality. In addition to the 2 clinical trials that I just described, there were also 2 meta-analyses of experimental therapies for COVID-19, and these provided additional information about the efficacy of remdesivir. The first one, the one that I am presenting on this slide, is a meta-analysis of remdesivir clinical trials and did not show statistically significant mortality benefit for remdesivir. However, there appeared to be a trend towards a lower mortality with remdesivir among those who were not on mechanical ventilation at baseline. On this second line, I'm presenting a second meta-analysis that had a broader focus, which means it was not only focused on remdesivir, and was a living systematic review and network meta-analysis that compared the effects of investigational drug treatments. So not only remdesivir, but other investigational drug treatments for COVID-19. This network meta-analysis was recently updated and published in December 2020. Studies of interest were randomized clinical trials in which people with suspected, probable, or confirmed COVID-19 were randomized to drug treatment or to standard care or placebo. The data sources for this meta-analysis were the World Health Organization COVID-19 database, which is a comprehensive multilingual source of global COVID-19 literature, which merged with the United States Centers for Disease Control and Prevention, the CDC, COVID-19 research articles downloadable database in October 2020, and 6 additional Chinese databases. There were over 20,000 titles and abstracts and 370 full texts screened for this analysis. 130 unique randomized controlled trials from 118 publications were identified, including 25 trials enrolling over 41,000 patients, which met the inclusion criteria. Several of these trials could not be included in the analysis for various reasons. The 43 of the remaining 75 trials, which had at least 100 patients or 20 events, were analyzed and formed the basis for this most recent updated publication in December. The conclusion of this network meta-analysis was that corticosteroids probably reduce mortality and mechanical ventilation, and the need for mechanical ventilation in patients with COVID-19 compared with standard care. Azithromycin, hydroxychloroquine, interferon beta, and tocilizumab, an interleukin-6 inhibitor, did not reduce either. The clinical benefit of remdesivir remains uncertain. So now we have differing information from clinical trials and meta-analysis and different recommendations. The FDA has now licensed remdesivir, and remdesivir is indicated for the treatment of coronavirus disease 2019 for those patients requiring hospitalization. Whereas the World Health Organization states that remdesivir is not suggested for patients admitted to the hospital with COVID-19, regardless of how severely ill they are, because there is no evidence currently that remdesivir improves survival or the need for ventilation. Let's talk now about corticosteroids, the second group of drugs for which there is evidence and it's widely used for treating COVID-19. As I mentioned earlier, corticosteroids were the only drugs noted to reduce mortality in the Living Systematic Review and Network Meta-Analysis that compared the effects of investigational drug treatments for COVID-19. I would like now to discuss a very important study, the randomized evaluation of COVID-19 therapy, also known as the recovery trial. The recovery trial was a United Kingdom-based study that reported its findings from 6,425 patients randomized to 6 milligrams per day of dexamethasone or usual care. Patients were also allowed to receive other COVID-19 specific therapy, including lopinavir, ritonavir, hydroxychloroquine, azithromycin, convalescent plasma, or tocilizumab. Overall, as you can see here, dexamethasone resulted in an absolute reduction in mortality of 2.8%. This next slide shows that the benefit of corticosteroids was greatest for patients who were receiving invasive mechanical ventilation at the time of randomization, with mortality with an absolute risk of death reduced by 12.1% from 41.4% to 29.3%. No survival benefit was noted for those who were receiving no respiratory support at baseline. Looking at the data a different way, corticosteroids confer a survival benefit, but only for those who are receiving respiratory support. The benefit is the greatest for those who are mechanically ventilated. The results of the recovery trial led to the suspension of most ongoing trials of corticosteroids because the results were so good that other trials were suspended. However, a prospective meta-analysis of 7 randomized clinical trials of corticosteroids that included over 1,700 critically ill patients with COVID-19 was conducted and provided additional information regarding the use of corticosteroids in this population. On this slide are the clinical trials that were included in this meta-analysis. The following observations were made from this meta-analysis. Administration of corticosteroids was associated with a lower all-cause mortality at 28 days after randomization. The association between corticosteroids and lower mortality was stronger in patients who were not receiving vasoactive medication at randomization. The association between corticosteroids and mortality appeared similar for older and younger individuals, men and women, and for longer and shorter durations of symptoms before randomization. Importantly, there was no suggestion of an increased risk of serious adverse events as a result of corticosteroids. Given these results, the NIH and the World Health Organization both recommend the use of dexamethasone in patients with severe COVID-19 pneumonia. The recommendation is stronger for those who are mechanically ventilated. Dexamethasone is not recommended, or corticosteroids are not recommended for those who do not require supplemental oxygen. I will finalize my presentation with a few comments on vaccines directed towards COVID-19. This obviously is a hot topic. I just received my second dose of COVID-19 vaccine this morning, and I'm very fortunate to be able to do so. First, let's discuss the usual process and timeline for vaccine development. Development of vaccines is an arduous and multi-step process that includes research and development, involving platform selection, designing targets, preclinical testing in vitro, in cell culture, and in vivo testing in animals, obviously. If this preclinical testing produces encouraging results, then the vaccine candidate is evaluated in human volunteers through clinical trials. These clinical trials are conducted in 3 phases, but some of these phases can be conducted simultaneously if the epidemic situation warrants it, which is the case in the COVID-19 pandemic. The work of making vaccines available for clinical use does not end when vaccine safety and efficacy is demonstrated in clinical trials and regulatory approval is obtained. A major coordinated logistics operation that involves not only the manufacturing aspect of things, but supply chain distribution and storage has to be undertaken. And you are very aware of the importance of this issue through all the attention it has gained in the news recently. As can be expected, this process takes a long time. For example, it took almost 60 years from the time of the first polio outbreak in the United States to the development and availability of the polio vaccine. In recent years, we have become a bit better at this, and the development time frame has been shorter. For example, it took only 15 years for the development of the Ebola vaccine, and 17 and 16 years for the development of vaccines against SARS-CoV-1 and MERS. However, the development of the vaccine against COVID-19 is unprecedented in that never before seen data sharing and collaborative efforts across scientists, pharmaceutical companies, organizations, and countries have managed to develop and deliver effective and safe vaccine in less than a year. Let's now discuss briefly the vaccine platforms. A variety of platforms and approaches are utilized in the development of vaccines against COVID-19. I will briefly describe those, and I also will attempt to give examples of vaccines in current clinical use that use these platforms. First, let's talk about nucleic acid platforms. There are two nucleic acid platforms, DNA and RNA-based. Note that these nucleic acid platforms are relatively novel platforms. There are no currently licensed vaccines that use these platforms. The DNA-based vaccines work by inserting synthetic DNA of viral genes in two small DNA molecules. The cells take in the DNA plasmids and follow their instructions to build viral proteins, which are then recognized by the immune system and prepared to respond to disease exposure. The RNA vaccines, and we will talk more about these later on, introduce an mRNA, messenger RNA sequence coded for a disease-specific antigen. Once this antigen is reproduced within the body, it is then recognized and triggers an immune response. The next vaccine platform I will be discussing are the protein-based vaccines. These are also known as subunit vaccines. These vaccines introduce a fragment of the virus, an immune-stimulating viral protein antigens into the body. This fragment is enough to be recognized by the immune response and to stimulate immunity. The protein-based vaccines contain the viral proteins, but not genetic material. Some contain whole proteins and some contain only fragments of them. An example of a subunit vaccine is the influenza vaccine. Viral vector vaccines insert a viral gene or a gene for a viral protein into another harmless virus, which delivers the viral protein to the vaccine recipient, and then triggers an immune response. Some viral vector vaccines enter cells and cause them to make viral proteins, and these are called non-replicating. Other viral vectors slowly replicate, carrying coronavirus proteins on their surface, and these are called replicating viral vectors. Live attenuated vaccines are made up of whole viruses that have been weakened in a lab. and they tend to elicit a stronger immune response than inactivated vaccines. Examples of currently used live attenuated vaccines include measles, rubella, mumps, yellow fever, and smallpox vaccines. Inactivated vaccines consist of the whole virus, which has been killed with various techniques, including heat or chemicals, so that it cannot cause illness, but will elicit the immune response. Examples of currently licensed vaccines, which are inactivated vaccines, are the rabies vaccines and the hepatitis A vaccines. Examples of vaccine candidates for against SARS-CoV-2, against COVID-19, and the various platforms that they are utilizing is provided on this slide. The pipeline for vaccines against COVID-19 is very robust. The World Health Organization maintains a twice-weekly updated COVID-19 candidate vaccine landscape database that compiles detailed confirmation on COVID-19 vaccine candidates in both clinical and preclinical development. As of December 29, 2020, there were 172 vaccine candidates in preclinical development and 60 vaccine candidates in clinical development in this database. These candidate vaccines utilize different vaccine platforms and also differ in their dosage, dosing schedule, and route of administration as detailed on this slide. Another resource for tracking the progress of COVID-19 vaccine is the New York Times Coronavirus Vaccine Tracker. As of January 4, 2021, 64 vaccines are in clinical trials in humans. 20 of those have reached the phase 3 advanced clinical evaluation stage. This next slide details the leading vaccine candidates, their developers, the platform that they are using, and their regulatory status. In the United States, 2 vaccines have received emergency use authorization from the Food and Drug Administration. The 2 vaccines are the BNT162b2 from Pfizer and BioNTech, and the mRNA 1273 vaccine from Moderna. I will highlight some of the characteristics of these 2 vaccines. The vaccine that I personally have received is the Pfizer BioNTech vaccine, if you are interested. As I stated previously, mRNA, the messenger RNA vaccines, represent an entirely new vaccine approach. mRNA is the intermediate step between the translation of protein encoding DNA and the production of proteins by ribosomes in the cytoplasm. Naked mRNA is quickly degraded extracellular. To avoid this and to enhance its delivery into cells, it is encased in lipid nanoparticles. Once administered, the mRNA is translated into the target protein, intended to elicit the immune response. Advantages of an RNA or a messenger RNA vaccine platform include that the mRNA remains in the cell cytoplasm. It does not enter into the nucleus. It does not interact with or integrate into the recipient's DNA. mRNA vaccines elicit broad immune responses, and the technology enables the rapid development, production, and scale. So these are great advantages. Let me talk about the BNT162b2 vaccine. The BNT162b2 phase 2/3 clinical trial enrolled a little over 43,500 participants, 16 years or older, and randomized them one-to-one to receive vaccine or placebo. The baseline characteristics of these two groups are comparable and are presented here on this slide. In terms of efficacy, there were 8 cases of COVID-19 among the vaccine recipients and 162 cases among those assigned to placebo, which means BNT162b2 was 95% effective in preventing COVID-19. When they looked at various subgroups, such as age, sex, race, ethnicity, baseline body mass index, and the presence of coexisting conditions, the vaccine efficacy was similar. The vaccine had a good safety profile. More BNT162b2 recipients than placebo reported adverse events, 27 versus 12%. The most common adverse events were short-term and mild to moderate, pain at the injection site, fatigue, and headache. The incidence of serious adverse events was low and was comparable between the vaccine and the placebo groups. Let me say a few words now about the second vaccine that has been authorized in the United States, the mRNA 1273 vaccine. The phase 3 placebo-controlled trial of the mRNA 1273 vaccine were recently published. This trial was conducted at 99 centers in the United States and enrolled over 30,000 subjects with over 15,000 participants in each group. That is vaccine versus placebo. The baseline characteristics are provided on this slide and were comparable between the two groups. Here is the efficacy data for the mRNA 1273 vaccine. COVID-19 was confirmed in 185 participants in the placebo group and in 11 participants in the mRNA 1273 group. This translates to a vaccine efficacy of 94% in preventing COVID-19 illness, including severe disease. When they looked at subgroups, the efficacy was similar across these groups, including age, gender, race, and ethnicity. What about safety? As regards safety, there were local reactions to the vaccination that were mild. There were also moderate to severe systemic side effects such as fatigue, myalgia, arthralgia, and headache. These were noted in about half of the participants after the second dose and started about 15 hours after vaccination, and in most of the participants resolved by day 2 without any further sequelae. And again, these two vaccines had very amazing high level of efficacy with mild and acceptable safety profile. And with this, I conclude my presentation. I hope that the information that I have provided was useful. I thank you for the opportunity and for your attention. I look forward to future interactions, discussions, and collaborations. Keep safe, be well. I wish you all a healthy 2021.